Oh, yikes, this is the back of the slideshow. Welcome to chapter 11. So this is like in reverse. So I don't want to spend too much time. This dude is Gregor Mendel. He was a monk in Europe, and he did experiments with peas, sort of like I did experiments with moss. And what Gregor Mendel found is that when he bred true breed pea plants that had purple flowers with white flowers, all of their offspring, F1, was purple flowers. You've probably heard this story before, so I don't want to spend too much time on it. And what Mendel was doing is what we call hybridization, where we take two individuals of the same species that can have successful reproduction, and we breed individuals that have different unique traits. We do this with cows to get certain types of traits within cows in terms of their butter fat content in their milk. We do this with many types of reproducing vegetables and fruits to get certain types of traits or cultivars. Think of blueberries or peppers. And it's a way for us to look at how traits are passed from one generation to the next. The true breeding parents that are sort of like non-hybridized, we always call the P generation. Their hybrid offspring are called the F1. And then when we take F1 generations and we have cross-pollination occur, that is what we call the F2. And when Mendel did this, he found that, lo and behold, about 1/4 of all F2 individuals were white. They were expressing what we would now call the homozygous recessive allele in their phenotype. And when Mendel did this with lots of different P traits, whether it's flower color, seed shape, pod color, flower position, stem length, he saw basically the same 3-to-1 ratio. And you could do this at home if you wanted to plant a garden full of peas from year to year. Now, Mendel used a simple mathematical model to help explain the 3-to-1 inheritance pattern that is actually founded in later observations of mitosis and meiosis. So an interesting thing to consider is Mendel did all of this work without the images that we now can see of cells doing meiosis and mitosis. The first of the 4 concepts that he created was the idea of alternate versions of genes account for inherited characteristics. Basically, there are different variations of genes on chromosomes that are expressed to make proteins, and those proteins, based on the genetic code, have slightly different phenotypes or physical expressions. Think blue eyes versus brown eyes, curly hair versus straight hair, dimples or non-dimples. These are genes that are being expressed that have slightly different genetic codes in terms of the nucleotide sequence, the codes for the amino acids that make our tertiary and quaternary protein structures that are based off of R groups. Yes, I just went into a chapter 3 deep dive. And alleles are found on the homologous chromosomes, dad and moms, with variations in them. The second variation is that an organism inherits 2 alleles, one maternal and one paternal, and this is due to the separation of homologs during meiosis in your mom and dad, who are separating themselves their maternal and paternal genotype. So in a sense, you are a result of the division of your grandparents' genes within your mom and dad's reproductive organs, making some gametes. And then the third idea is that if the 2 alleles at a locus differ, then one of the allele is going to be dominant and the other is recessive. The idea is that one allele masks or outcompetes generally the other allele in terms of the physical expression, sort of like David versus Goliath. One of the alleles is going to come out on top. And in the flower case, the pea plant allele was clearly the purple dominant allele because it masked the white allele. And the last idea is the law of segregation. The 2 alleles for a heritable characteristic sort of separate during gamete formation. And we know this because of independent assortment and the division of homologs during meiosis 1. So we might get mom or dad genes depending on how that process goes. If an organism has 2 identical alleles, we call them homozygous, homo meaning same or similar. And if an organism has 2 different alleles for a gene, we call them heterozygous, or hetero meaning different. heterozygotes are not quote-unquote true breeding. And this is very important in cases of organisms where pedigree matters quite a bit, like purebred golden retrievers. We talk about true breeding golden retrievers if they have 100% of the alleles that we would expect to see in a golden retriever breed. And because of dominant and recessive alleles, an organism's traits do not always sort of reveal its genetic composition. So that's why we talk about phenotype and genotype, because many individuals can be carriers of a recessive allele and be heterozygous, but then physically they look identical to a homozygous dominant phenotype. And this is where we can sort of see that difference between a phenotype and a genotype. Now, we can use test cross to sort of understand the unknown genotypic expression or characteristics of an organism by reproducing it or breeding it with a true bred recessive. And that will allow us to sort of understand what is the parental genotype of an individual. So if we test cross an individual that's homozygous dominant with a homozygous recessive, we would expect to see, based off of independent assortment of the alleles and complete dominance, 100% purple. But if this Papa P is heterozygous dominant, there's a 50-50 chance that some of the offspring will inherit a little P allele, so we should see half of the individuals being white. And we can do this also with a dihybrid cross, where we can see different ratios of wrinkled and smooth and yellow and green peas come out to often being a 9:3:3:1 ratio. We call this a dihybrid cross, where about 9 of the 16 individuals will be dominant for both traits, 3 for one and recessive for the other, vice versa, and then only 1 out of 16 should be recessive. This is just based off the idea of independent assortment of the homologous chromosomes on the metaphase plate during meiosis 1. And this, again, really helps us just think about how genes are separated and duplicated within gamete formation. And the probability is always just going to be based off what's the probability of inheriting one of these traits. So there are some inheritance patterns that are a little bit more complex. Not all of the heritable traits are just simply a complete dominance trait. One of them is incomplete dominance, where we sort of have the phenotype of F1s between the two parental varieties. Snapdragons that are pink from a parent that's red and white is a good example of this. And codominance is where the phenotype expresses both traits simultaneously and sometimes in separate and distinguishable ways. Blood type, if you're AB, is a good example of that. You aren't just A and you're not B, you have A and B antibodies on your blood. Here's a good example of the F1 and F2 generation. And interestingly enough, if you breed a pink snapdragon with another pink snapdragon, we start to see a higher expression of the pink than the red or the white because of that heterozygous inheritance pattern. There is a couple different types of diseases that we need to talk about. One is Tay-Sachs disease, which is fatal. It's a dysfunction in the enzymes that cause basically lipids to accumulate in your brain. At the organismal level, this allele is recessive. At the biochemical level, the phenotype is incompletely dominant, meaning that if you're a carrier for Tay-Sachs disease but you don't express it, you aren't going to actually express the trait, which means that you can pass it on to offspring. And at the molecular level, the alleles are codominant, meaning that both protein structures are being expressed simultaneously. The relationship between dominance and phenotype, so alleles are just simply variations in a gene's nucleotide, which makes proteins. When a dominant allele coexists with a recessive allele and a heterozygote, they don't really interact, but rather the two proteins are expressed, and then those proteins interact with cells and cell signaling and organismal tissues, and basically the result will be whether or not the traits of that individual are different than a true breeding dominant or recessive individual. For any character or gene, dominant and recessive relationships of alleles depend on the level at which we examine sort of the phenotype in terms of whether it is at the organismal level, biochemical level, or the molecular level. Dominant alleles are not necessarily more common in populations than recessive alleles. So for example, one type of dominant allele is when you are born with polydacty, which is an extra finger or toe, and that is caused by a dominant trait, but only 1 out of 400 babies in the United States are born with this trait. Most genes aren't just binary. There are many alleles that exist with 2 or more allelic formations. So for example, blood type can be IA, IB, or little I. So you can be type A, which would be 2 versions of this. You can be B, which would be 2 copies of this. You can be AB, or you can also be type O, which would be 2 copies of the little I. And we can see that right here in terms of blood type. This is why we say O is the universal donor, because it lacks the proteins on here, which means it won't be rejected by the immune system of anyone that is AB, B, or A. And AB is generally the universal acceptor because it can interact with B and A and I. But a type A person can only receive type A or O blood. If it receives the B or AB, there will be some mishap with the protein recognition, so the body will reject those cells. Geotrophy is the idea that there can be alleles interacting with other alleles to cause a phenotype to be changed. So for example, if you inherit cystic fibrosis, which is a recessive allele, there might be many traits expressed as a result of that one single allele being inherited. And the other notion of multiple alleles is this idea of epistasis, where a gene at one locus sort of changes the phenotypic expression of a gene at a second locus. And this is often because the first gene codes for a protein that is called a transcription factor, which may turn on or turn off the second gene by going to that chromosome and interacting with that gene. So for example, the allele B codes for whether or not you are a black or brown lab. So cute. And the allele E codes for whether or not that pigment is expressed. So this B, B, E, E golden lab really ought to be a black lab, but because it lacks the second allele for expressing that pigment, it ends up being golden. And the same thing is true for this should be brown retriever, little b, but because it has those little E alleles, it doesn't code for the pigment. And we can also consider quantitative characters. Quantitative characters are those that vary in a population sort of along a spectrum, and this generally is based off the fact that there's polygenic inheritance going on. There are more than one allele coding for the physical expression that we see. Skin color is a good example of this, where we can have dominant and recessive alleles for the gene A, B, and C, and then generally based on the amount of dominant or recessive alleles that we see, the skin color of an individual sort of fall in a continuity. And by no stretch of the imagination, most individuals, based off of a Dihybrid Cross, big A, big B, little b, big C, little c, will end up sort of in the middle of that spectrum. Another departure from Mendel genetics in terms of just simple binary dominant recessive alleles is also the idea that a phenotype or a character depends on the environment as well as a genotype. We call this epigenetics, where the phenotypic range of a genotype might be influenced by things like carcinogens or water salinity or pH or soil temperature. It's not just genetic inheritance, it's other things that might impact how you physically express a trait. Now we can analyze whether or not a trait is being passed on from generation to generation within a pedigree, which sort of shows parents and children across generations, and the patterns of traits that are being sort of examined. So for example, we can look at widow's peaks versus no widow's peaks, and this is a very good example of a dominant trait because each generation of reproducing individuals display the trait. There's no skip generation. Here, an individual gets an attached earlobe, little f, despite having two parents that don't have attached earlobes. And this is a good indication that it's a recessive trait because the two parents must be carriers if they don't express the trait. A good example of a recessive allele is cystic fibrosis. About 1 out of every 25,000 people of European descent is born with it. And the allele results in defective or absent chloride transport channel proteins in the plasma membrane, which basically means that chloride ions build on the outside of the cell. And that causes mucus to build up on organs and tissues in the lungs, which really prevents absorption of gas and nutrients in the small intestine and in the lungs. It is lethal. It does shorten life expectancy by 30 to 40 years. And sickle cell anemia is also another recessive allele that's surprisingly common. 1 out of every 400 African Americans have the disease, and it is caused by just a single amino acid substitution in the quaternary structure of hemoglobin. And homozygous individuals that are recessive have sickle cell cells. And the symptoms can be physical weakness, pain, organ damage, even paralysis. And finally, there are some dominantly inherited disorders that are caused by dominant alleles. And dominant alleles generally cause lethal disease that is a rare thing. It generally prevents successful reproduction, and it's often spread by two carriers reproducing that might not have a full dominant expression of the trait. And we can see that with dwarfism, which is known as achondroplasia. I'm going to really screw this up. Achondroplasia. There we go. Where we have a dominant allele interacting with normal alleles, causing a 50-50 expression. Now, there are some dominant alleles that we ought to expect to be bred out of existence, so to speak, based off their implications on the phenotypic expression of an individual. Huntington's disease is a good example where a degenerative disease of the nervous system occurs. Sounds pretty lethal to me. But the problem is that the disease has no phenotypic effects until about 35 to 45 years of age, at which point people might have had 1, 2, 3, 4 kids. So it has been something that is really important to screen for in terms of genetic testing to make sure that people don't unknowingly pass on a trait to future generations. I hope this has been helpful in terms of Chapter 11's overview. I will see you guys tomorrow. Have a good